just to sort of refresh on some terms that I'll be using throughout the presentation. After that, I'll speak about why this topic is specifically important in our field and just provide a basic understanding of genetics. I'll then do an overview of various genetic studies. The bulk of the presentation, however, will focus on clinical manifestations of inherited childhood conditions, and we'll end off with some information on genetic testing and counseling and what is currently available to us. So I'm not going to embarrass anyone. This is just for our own revision, so you can test yourself. So this is the start of the quiz. Genetic testing is advisable in the case of children with ADHD, helps to choose the right treatment for a specific child, not advisable for most common child psychiatric conditions. In most cases, genetic testing eases parental anxiety, or genetic testing is available in most countries nowadays. So I'm sure the medical students are keen, but so the answer to this is C. It's actually not advisable for most common childhood psychiatric conditions. So the next concept relates to epigenetics, which is very topical in our time. What does it refer to? Is it A, changes in the organism that are outside of the cell? B, evolution due to natural selection? C, changes in RNA protein binding? Is it heritable changes that do not involve changes in DNA sequence, or a field of study that encompasses intracellular mutations? I can say that when I was at medical school, I don't recall a single lecture on this topic showing how recent the developments in epigenetics are.
And Ilhaam, I'm going to suggest, I was going to say, let people type the answer in the chat box. Never mind, I'm too late.
Okay, sorry. Okay, we can do that. There's 2 left, we can do that. So, sorry, that is the same slide. So, what does mitosis refer to? I'm going to give you a minute to read it, and then you can type the answers in the slide, in the chat box. I'll be interested to see.
Maybe you could just read it out loud.
Okay, so I'm not sure if anybody answered. I see there's a few answers, but the correct answer is A, a cellular process that replicates chromosomes and produces 2 identical nuclei in preparation for cell division. So the last test in the quiz is, who was Gregor Johann Mendel, after whom the concept of simple or Mendelian genetics originated from? What was his occupation? Was he a poet and philosopher, friar and abbot, pilot and farmer, doctor and scientist, or factory worker? Not sure if any of you know, but the correct answer is B. He was a friar and abbot. So well done to those of you who got it correct.
Had some good answers.
So why choose this topic? Yeah. Why are genes relevant in psychiatry? So advances in behavioral and molecular genetics have benefited us greatly over the past 20 odd years. Child and adolescent conditions were once assumed to be caused by a myriad of reasons, including, as you may know, fixation on certain periods, what were deemed refrigerator mothers, so-called deviant behaviors. The discovery of genetics has made it less fashionable to argue against biological underpinnings of developmental psychopathology. So most of us understand genetics in terms of single gene, or what is referred to as simple or Mendelian patterns of inheritance. To recap, that refers to sort of autosomal dominant and recessive traits, and this was a picture of Gregor Mendel, who developed that concept. However, child and adolescent psychiatric illnesses are prime examples of what is referred to as complex traits. What this implies is that it is associated with multiple genetic and environmental triggers acting together. So just a basic recap of concepts. We all know that each cell in the human body, with the exception of red blood cells, contains 23 pairs of chromosomes. We inherit these from our parents, 23 from your mother, 23 from your father. And chromosomes are made up of tightly coiled strands of DNA. This is a graphic representation of DNA. When uncoiled, this double helix pattern, which is in the shape of a twisted ladder, it reveals what is known as chromatids, and they are chemical bases. I don't know if any of you should recall the 4 basic bases, which is adenine, thymine, guanine, and cytosine, which form interlocking pairs. And the order of the specific ladder is referred to as our DNA sequence. This concept is important for us in child and adolescent psychiatry, as DNA is passed on from parent to child, and then each DNA contains one strand or one chromatid from one parent and a second strand or chromatid from the other parent. And these strands of strings are what is referred to as nucleotides. They cluster into functional units, what we refer to as genes, which code for specific proteins. So genes often come in 2 forms called alleles. And a child receives one allele from their mother and one from their father. Thus, if one were to determine that specific alleles of a gene follow along with a particular disorder within a family, one could then derive associations between specific genes and specific disorders. So the building block, or what is referred to as the central dogma of biology, is that DNA, via a process called transcription, changes into messenger RNA, and via another process called translation, changes into proteins. And proteins are the workhorses of cells. And this concept is central to psychiatric genetics. So the proteins are able to transport in and out of the cell membrane to serve as receptors for neurotransmitters. And specifically for us, with regards to psychopharmacology, most psychiatric drugs work via neurotransmitters. This changes the way in which the cell fires and therefore exerts an effect on biological systems. And because proteins in neurons start first as basic code in DNA, the way that neurons function can be affected by the variation in the DNA code. So DNA taken from any cell in the body, and typically if any one of us were to go for a DNA test, they take it from lymphocytes, from lymph glands or salivary glands, can provide insight into the DNA sequence throughout the rest of the body. So if a change in the DNA code is associated with a psychiatric condition, one would be a step closer to understanding biological underpinnings of that condition. Oh, so sorry, I don't know what's happening. It reflected better on my screen. But basically, this is just a graphical representation that DNA is a double-stranded molecule, as you can see here. Again, to recap the technical jargon that I referred to earlier. One strand comes from your mother, the other from your father, and on each of these strands or chromatids are strings of nucleotides, which cluster into functional units or genes that code for proteins, and genes come in two forms called alleles. So this is a picture of a chromosome. Each one is a chromatid. This is the cell. DNA is situated in the nucleus of the cell, and this represents the actual proteins. So again, a child receives one allele from his mother, one from the father, and the alleles contribute to the organism's phenotype. Phenotype refers to the outward appearance of the organism, and that is why we all look different. Some of us have black, brown, or blonde hair, some of us are tall or short. And because half of the DNA of an individual is transmitted from the mother and the other half from the father, this has enabled us to determine specific inheritance patterns for particularly single gene disorders. An example of this would be Huntington's chorea, which is inherited due to an expansion in the number of specific trinucleotide repeats of a particular area on the genome. Okay, so that was a bit technical. Just to say, in terms of the hypothesis for genetic disorders, there are three sort of schools of thought in child and adolescent psychiatry, and they're not mutually exclusive. So we'll briefly review them. The first hypothesis is called the common disease, common variant. What this says, so this hypothesis says that child psychiatric disorders are due to the accumulation of multiple common genes, each which have a minor additive effect on the presentation of the illness. In this hypothesis, genes which may otherwise have been selected against through natural selection are kept in the population, because in other environments and in combination with certain other genes, they are advantageous. However, if we cluster enough of these risk alleles together, they can push a child over the threshold and they can manifest as an illness. Support for this hypothesis comes from findings related to studies done on autism spectrum disorder and ADHD. And a scientist called Constantino showed that subsyndromal autistic traits are found in siblings of individuals with autism. And similar arguments have been made for ADHD. What this suggests is that if, for example, we were to see a 10-year-old female patient with autism, chances of their siblings having subsyndromal symptoms is higher than the general population, according to the specific hypothesis. So this suggests that there's a familial aggregation of genes that affect the individual, but some individuals have a higher dosage than others. Another hypothesis is something called the rare variant common disease. So what this says is that common psychiatric disorders are associated with an accumulation of rare mutations in a population. And this would explain why some people who have no family history of, for example, schizophrenia, could have a family manifest with this illness. And proponents of this hypothesis postulate that one or several of these rare, which is less than 1% of the allele frequency mutations, placed along a critical development pathway may push the child towards manifesting this disease. And this has been useful to explain disorders where there is the appearance of a new condition in an otherwise healthy family member. And this process is referred to as de novo mutation. In the rare variant common disease hypothesis, they become problematic when these de novo mutations accumulate. So, for example, if rare variants are overrepresented in autistic families where there is a single autistic individual, because there are new mutations that can be passed along, they have profound effects on each subsequent generation, which makes the gene pool, the likelihood of future generations developing or manifesting a specific illness higher. And at the extreme, in other words, over multiple generations, if a high gene pool of mutations develop, the equivalence can almost look like typical or simple Mendelian traits, where your chance of developing an illness is high. Then, of course, we all know about the gene-environment interaction. So this is, as the name suggests, it is assumed that either common variants or rare variants only have an effect on the expression of child psychiatric conditions if placed into an environment in which those particular genes were expressed and could therefore be deleterious. So in the early 2000s, two scientists wrote an influential paper proposing that the type of environment in which a child is raised placed them at a higher risk for later illness. And this is particularly of interest to us, being in a low socioeconomic setting or having quite stressful environments. So depending on the child's genotype, but it says that the genes are not enough to manifest the illness. That is what this hypothesis postulates. And the findings of these two scientists sort of laid the foundation for epigenetics and gene expression in child and adolescent psychiatry. So there's various studies that we use to, when working with genetic disorders. The sort of bread and butter would be family studies. So what this implies is that DNA, as we know, is passed from parent to child. Therefore, disorders in which genes are sorry, in which, struggling to see this. So in which genes are passed down. This is useful in establishing heritability of almost all child psychiatric conditions. And family studies require information from at least 2 sources. Obviously, the more extensive your family tree, the more genetic information you'll have at your disposal. Another type of study is twin and adoption studies. And these are useful. Although adoption studies are rare, they've provided marked insight into environmental factors that influence strays. In adoption studies, children are assessed to see whether they are more similar to their birth parents or to their adoptive parents. And if the children resemble their adoptive parents more than expected, this indicates shared environmental influence. Adoption studies, while less common than either twin or family studies, have demonstrated shared environmental associations, specifically for antisocial behavior and personality disorders. So twin studies are useful because, in contrast to adoption studies where the environment is varied, twin studies allow separation of the shared environment. So you get 2 types of twin studies, those relating to identical monozygotic twins and those relating to dizygotic or fraternal twins. One would assume at the get-go, and this is an oversimplistic explanation, but that in the case of identical twins, they share 100% of their genetic sequence, versus adopted twins, the rate is, versus non-fraternal twins, that genetic coding is deemed as 50%. And this is useful because it's useful to exclude or include the relevancy of genes and disorders. There's also something called candidate gene studies. And once family studies or twin and adoption studies have determined the degree of heritability, researchers can investigate specific genes involved. So that is essentially what candidate genes implies. So this looks at the specific genes hypothesized to be involved in the presentation of the illness. And this is useful for us because it allows us to unlock physiological pathways important in the development of genetic disorders. There's also something called linkage studies, which some of you may have heard of, and this just refers to, this is a specific study that uses the entire genome, so it determines the specific location on the chromosome that a disease may lie. There's also something called GWAS, or genome-wide association studies. And what this does is, it is a genome-wide set of genetic variants in different individuals to see if any specific variant is associated with a trait. And to recap, the definition of a genome is the complete set of one's DNA, including all your genes. A GCTA, or genome-wide complex trait analysis, is an alternative way to use whole genome data to examine the additive effects of genes directly. And then we have epigenetics, as was previously mentioned, and epigenetics provide an additional approach to psychiatric genetics. So it's derived from the Greek word, which literally means outside of genes. It was first used to describe heritable changes in the genome that are not related to changes in the DNA sequence. So it's basically things which are outside of the cell, which can switch genes on and off, for certain proteins, etc. Then, as mentioned also before, the gene and environment interplay. It has long been known that genes and environment interact with one another. Just as it could be argued that genes and the environment lay the foundation for natural selection, organisms whose genotypic expression allows them to survive in the environment in which they were placed will be able to reproduce, passing on their genes to the next generation. And this concept is at the forefront of psychiatric genetics. According to Yakupap, which is our prescribed textbook, the old adage of nature versus nurture is almost always believed to be nature and nurture working towards the expression of child mental health presentations. So, nearly all of the common child and adolescent psychiatric conditions or their related constructs have found to have genetic components. While this may seem like a strange statement, it is noted that not a single DSM-5 condition listed in the childhood disorders section does not have heritability as a component. In other words, the etiology is postulated of every single known child and adolescent psychiatric condition to owe some of it to genetics. So that brings us to the question, what is heritability? So this refers to the proportion of observed variation in a particular trait, such as weight or height, sorry, not weight, but height, that can be attributed to inherited genetic factors, in contrast to environmental ones. It's a statistical sample that describes how much of the variation in a given trait can be attributed to genetic variation. And this can include characteristics such as eye color, intelligence, height, as well as disorders like autism and schizophrenia. So essentially what this implies, and Prof, please correct me if I'm incorrect, but for example, with conduct disorder, it's not done on an individual basis. This is population-based studies, which of course is not finite, but that 39 to 63% is postulated to be due to genetics. That is what it implies. And the rest, so in other words, a maximum of 61% is attributable to the environment with regards to conduct disorder specifically. So the next aspect of the presentation, we're done with all the very loaded theoretical information, will be specifically clinical significance of this topic to us in child and adolescent psychiatry. So for the medical students, without even looking at the title, you'll recognize this baby as having Down syndrome. So Down syndrome describes a constellation of symptoms associated with 3 copies, and that is what trisomy refers to, of chromosome 21. So the genetic name of Down syndrome is trisomy 21. It's often detected during prenatal screening and has been associated with both advanced maternal and paternal age. Trisomy 21 occurs in about 1 in 1,000 live births, and that parents most often have unremarkable karyotypes. So the additional genetic material from the extra chromosome results in this very specific cognitive phenotype. There are non-psychiatric and numerous psychiatric and behavioral manifestations of Down syndrome. It has very specific or distinguishing facial features, which the medical students will know, which includes upslanting palpebral fissures, which is sort of the upslanting of the eyes over here, epicanthal folds, which is also referring to the eyelid region, a broad nasal bridge, small mouth, relatively large tongue, posteriorly rotated ears, short stature. And if you all look at our hands, we all have numerous creases on our palms, and characteristically, children with Down syndrome tend to have a 1 palmar crease. There's also numerous physical associations outside of the facial features, including cardiac anomalies, GIT manifestations, and numerous neurological and psychiatric comorbidities. It's important to note that most individuals with Down syndrome do in fact have intellectual disability. Language delay is common. While social responsiveness is not necessarily impaired, up to 10% of individuals with trisomy meet the criteria for autism. And patients are at increased risk of many child psychiatric disorders and neurological problems, including ADHD, anxiety, depression, epilepsy, and have a high incidence of developing dementia later in life. I don't know if anybody can recognize this syndrome. Just from the picture, it's not very, I don't know why, my slides aren't projecting well on Zoom at all. This is something called Fragile X syndrome. And in contrast to trisomy trisomy 21, which typically occurs as a de novo mutation in the germline of the parent, or a rare mutation, Fragile X syndrome is inherited from parent to child. This is associated with intellectual disability, and the rate or the incidence of this is about 1 in 4,000 live male births and 1 in 8,000 live female births. There's a reason for that, and it's believed that because one X chromosome is inactivated in the cells of females, females are less affected. The pathogenesis of Fragile X syndrome involves increasing number of trinucleotide repeats, and this results in a specific gene on the X chromosome being mutated. Across generations, more repeats accumulate, and when they accumulate, it's thought that you need higher than 200 repeated accumulations for the clinical syndrome to emerge. I don't know if anybody recognizes this, this syndrome, but this is referred to as Williams syndrome. So Williams syndrome occurs in 1 in 10,000 live births. It's related to deletion in a region on chromosome 7, and it affects around 30 genes involved in development. There are numerous non-psychiatric and psychiatric manifestations, and children with Williams syndrome have characteristic facial features. I'll take you back to the previous slide. So bear in mind, the facial features are a broad forehead, wide mouth, full lips, and a narrowing of the distance between the temples. I think what's captured quite nicely on this photo is that from a psychiatric perspective, they are known to be quite a hypersocial. They have a superficial hypersocial personality. Also, in terms of physical features, there's an increase in joint problems and elasticity of the skin and joints. And they may be associated with cardiac anomalies, specifically aortic stenosis. From a psychiatric and a developmental perspective, they typically have mild to moderate intellectual disability, especially visual spatial deficits, although the auditory rote memorization in language may be spared. And as mentioned, they have a superficial hypersocial personality, which may lead one to miss subtle social cues. ADHD symptoms are common, as are anxiety disorders and sleep disorders. And we can diagnose it by something called a FISH or microarray. So this baby suffers from a condition called Prader-Willi syndrome, and it is one of 2 conditions associated with the deletion of chromosome 15. Prader-Willi syndrome most frequently occurs, I think, when there is a deletion of the paternal copy and the maternal copy of the chromosome is inactivated. It occurs in 1 in 15,000 live births. Non-psychiatric clinical characteristics tend to appear early on in infancy with poor feeding, slow growth, developmental delay, and hypotonia. In children, hyperphagia becomes common with resultant obesity and the development of diabetes. There are characteristic facial features with a narrow forehead, triangular mouth, and almond-shaped eyes. Growth delay, particularly with small, narrow hands and feet, are common. Pubertal development is often delayed, and individuals have underdeveloped genitalia. So other psychiatric manifestations are common but not universal. They may have mild to moderate intellectual disability. And an interesting trait is that many individuals with Prader-Willi have compulsive behavior. Most prominent is hyperphagia or compulsive eating, which is thought to be related to the obesity and development of diabetes, but skin pricking, nose picking, and hair pulling are not uncommon. Interestingly, they do not have obsessions. They don't often occur. It can occur, but it doesn't often occur with the compulsions. Hoarding and gorging of food frequently occurs, as does poor frustration tolerance when limits are put on eating. Emotional regulation difficulties may also continue into adulthood. I don't know, this is a cryptic clue for an illness called Angelman Syndrome. So the sort of mirror image of Prader-Willi is something called Angelman Syndrome. It affects 1 in 15,000 live births. In this case, it also affects chromosome 15, but there is a loss of the maternal copy of a specific gene on this chromosome. And non-psychiatric manifestations include seizures, which often emerge in the first 2 years of life. There may be skin changes, such as hypopigmentation, coarse facial features that increases with age. Psychiatric manifestations include delayed development and intellectual disability. Children with Angelman syndrome appear happy and tend to laugh readily and frequently, at times associated with hand flapping. There's also social disinhibition, and sleep is frequently poor. To be honest, I'm actually not sure where the name is derived from. It will be interesting to know. So this girl has something called Rett syndrome, and the clue to her Rett syndrome is the positioning of her hands. So Rett syndrome is a mutation in a specific gene called the MECP2 gene. It occurs almost exclusively in girls at a rate of about 1 in 8,500 live female births. It can occur in males, but most males who have inherited this gene end up being mutated spontaneously. So non-psychiatric clinical characteristics are mildly delayed and typical early development for the first 6 to 18 months. Sorry, these are psychiatric clinical characteristics, is that it can be go unnoticed for the first year and a half. And what's characteristic about Rett syndrome is that despite the child seemingly appears well for the first 6 to 18 months, it is followed by a period of profound developmental delay, autistic traits, and the characteristic loss of purposeful hand movements. These hand movements are replaced with hand wringing, hand washing, or clapping movements. And I think this picture is trying to show a hand wringing movement. And unfortunately, there's eventual loss of motor function, and seizures are very common with this condition. And it often results in little or no language. Along with these hand movements, sleep difficulties can be present, as can irritability and autistic-like behaviors. This is a child with something called 22q11 deletion. And this occurs relatively frequently. Rates are estimated to be 1 in 4,000 people. And this incidence is thought to be underestimated due to the variability in the presentation. There's a loss of a specific gene, which is responsible for many of the non-psychiatric characteristics, called the TBX1 gene, whilst the COMT gene is thought to be due to the psychiatric manifestations. So the psychiatric clinical characteristics are variable, as are the physical characteristics. And some of them include, as indicated in this picture, a cleft lip and palate, other structural midline facial defects, cardiac defects, an absent thymus gland, hypocalcemia, and something called a conotruncal anomaly, which is a specific face syndrome. Some of the psychiatric characteristics of this is also, as mentioned, variable, but it may include borderline or mild intellectual disability, attention problems, anxiety, or social withdrawal, which can be present at a very young age. And many children with this illness do meet the criteria for an autism spectrum disorder. So this is a female with Turner syndrome, and it was described by Henry Turner as recent as 1938, and it was only 20 years later that the genetic basis of the syndrome was discovered. About 50% of those with this syndrome are associated with a single X chromosome, or referred to as X monosomy, and affected females have 45 rather than the usual 46 intact chromosomes. The prevalence of the syndrome, it occurs in females, is 4 per 10,000 live female births. This is the most common chromosomal aneuploidy, implying there's one less chromosome, although the vast majority of affected fetuses are aborted spontaneously. And in humans, partial or complete loss of either of the sex chromosome can result in this condition. There are many possibilities in terms of physical characteristics of this syndrome, if not detected at birth. And it's not that easy to detect it at birth. It is mentioned that it may be suspected when there's transient edema over the lower legs and feet, which rapidly clears. It often is made in middle childhood only. And often it's picked up when there is reason to be concerned about growth delay. Specific physical characteristics is identified in this picture relates to low set ears, a low hairline, occasionally a webbed neck. The eyes may show squints and slight ptosis. The chest is typically broad with widely spaced breasts, and when females stand with the arms at their sides, there's something called a wide carrying angle, which refers to the lower arms typically turn out at the elbows. It is also associated with cardiac abnormality, and that happens in about 1/3 of patients with Turner's. Other common problems include renal anomalies, and there's an increased risk of UTIs and hypertension. Interestingly, social integration is usually good until adolescence. The normal adolescent growth spurt is then lacking, and secondary sexual characteristics may be delayed until promoted by endocrinological management. So we manage this implementation. And as adults, many women with Turner's syndrome cannot function effectively in complex social work environments. And almost all have normal verbal intelligence. About 80% have relatively poor visuospatial memory. So what this is implying is that it can be quite difficult to recognize, and it can go undiagnosed, and the manifestations may be subtle, psychiatrically and physically. Social cognitive anomalies in Turner syndrome extend to deficits in mentalizing abilities. And in common with females who have what is referred to as idiopathic autism, girls with Turner syndrome attempt to compensate for their social deficits from early childhood. They may develop superficially good, engaging social skills, which are learned from imitation. So, in other words, they may deceive you into believing that from a social perspective that there aren't any concerns. But with time, the deficits may become more prevalent. And also associated with this could be the appearance of ADHD. This is something called Smith-Magenis syndrome, and this results from the deletion of a small piece of chromosome 17. And the deletion occurs on the shorthand of the chromosome, and this affects at least 1 in 25,000 individuals worldwide. Again, researchers postulate that the incidence is actually closer to 1 in 15,000 due to so many individuals going undiagnosed. So people with Smith-Magenis syndrome typically have affectionate, engaging personalities, but most concomitantly also have behavioral problems. These include temper tantrums, impulsivity, anxiety, and difficulty paying attention. Self-injury is also very common, including biting, hitting, and head banging, as well as something referred to as repetitive self-hugging, which is unique to this disorder. And some people with this condition also compulsively lick their fingers and flip pages of books and magazines, which is referred to as the lick and flap sign. So 16p11 deletion or duplication is a disorder caused by deletion of a small piece of chromosome 16. Typically, children suffering from this syndrome have developmental delay and intellectual disability. They may have also, in addition to that, features of autism spectrum disorders. They typically also have expressive language skills, which is usually the expressive language skills are usually more adversely affected than the expressive language skills. There's also minor physical abnormalities, which again can be difficult to pick up, including low set ears or partially webbed feet. And people with this disorder are also at increased risk of obesity compared to the general population. However, there is no particular pattern of physical abnormalities that characterize this deletion syndrome, and signs and symptoms of this disorder vary enormously among affected family members. And some people with this disorder may have no identifiable physical, intellectual, or behavioral abnormalities. So I had to mention this, and I've no idea why my slides are actually projecting so poorly on my Zoom, because it didn't project like this. Sorry about this on the presentation. But this is an interesting disorder that was first described by someone called Desiree Bourneville, a French physician in the 1880s. And he described the case of a young girl with severe intractable epilepsy. And based on, I'm assuming, post-mortem findings in the brain, they found potato-like growths in the brain. And this physician also described what he referred to as confluent vesiculopapular eruption on the nose, cheeks, and forehead. So genetics and molecular biology occurs as a spontaneous mutation in 70% of cases. The other 30% of cases are thought to be inherited via an autosomal dominant mechanism. And this disorder is associated with mutations in one of two genes referred to as a TSC1 and the TSC2, which occur on chromosomes 9 and 16 respectively. So the incidence of this is thought to be 1 in 5,800. There is a wide variability of expression. Historically, it was diagnosed using a triad of epilepsy, intellectual disability, and these characteristic skin lesions, which we see over here. But realistically, they are only seen, this triad is only witnessed in 30% of people with this disorder. It is a multi-system disorder and can include hematomatous growths, which is essentially benign growths which can affect multiple organs, including the brain, skin, kidney, heart, eyes, lungs, and even teeth. About 70 to 80% of sufferers of tuberous sclerosis do have a history of having suffered from epilepsy at least once in their lifetime. And the diagnosis is based on meeting clinical criteria. It's associated with a wide range of behavioral, psychiatric, intellectual, academic, neuropsychological, and psychosocial difficulties. And there's actually a screening tool which was developed on researching this, I discovered, by our very own Prof de Vries, called the TAND, which stands for the Tuberous Sclerosis Associated Neuropsychiatric Disorders. So it basically summarizes features that we should be looking out for. So it's a checklist which is available to us. At the behavioral level, tuberous sclerosis is associated with high rates of mood, anxiety, overactive, impulsive, sleep, eating, and dysregulated behaviors, which include aggression and tantrums. From a neurodevelopmental perspective, disorders are common, and it is thought that ASD is co-occurring in 40 to 50%. So almost half of cases of tuberous sclerosis manifest with ASD as well. And again, up to half of children with tuberous sclerosis may have ADHD. And the rate of co-occurring intellectual disability is also thought to be around about 50%. And I don't think I've highlighted that, but a co-occurring theme with these disorders is that there are numerous, not only physical, but psychiatric comorbidities, which are common to many of these chromosomal anomalies. And the tuberous sclerosis complex is one of the medical conditions most commonly associated with autism. And in adolescents and adults, very high rates of anxiety and mood disorders are reported. And I remember specifically learning about this in neurology as a medical student, where no emphasis was placed on the behavioral components such as autism and anxiety disorders. We're all quite familiar with the specific facial lesions because it's easily recognizable, but it's important to note that epilepsy can co-occur in up to 80% of cases, as well as autism, intellectual disability, and other psychiatric manifestations. So Klinefelter syndrome,
sorry, this is Prof here.
Yeah.
I just want to let you know we have less than 10 minutes left. So just so that it'd be nice if we can have a few minutes for questions at the end. So maybe you can just decide, well, I don't know how many slides you've got left, but sorry to butt in. I thought I should at least wait till after you've talked about tuberous sclerosis.
Okay, sure. Okay, thanks, Prof. Okay, so Klinefelter syndrome is interesting. I actually did have a patient that I'd seen with Klinefelter syndrome that I won't go into the technicalities of it, but it's classified as the most common sex chromosomal disorder, and this is sort of physical features of it. Interestingly, I think this is interesting to mention, last year, whilst at Gregor, I saw a 40-year-old male who presented with symptoms of ataxia, so he was unable to walk, and confusion. This was his first presentation to psychiatric services, and he was known to have no previous comorbidities and was previously well. On assessment, it was found that he had a B12 deficiency, which could account for both his ataxia and his confusion. Despite treating the B12 deficiency, his gait improved, but his delirium didn't. And it turned out that not only was he delirious, but that he had long-standing psychotic features in excess of 10 years, which presented very similar to the classical thinking of first-rank schizophrenia type symptoms. In hindsight, it was discovered that he had numerous autistic features, that he actually had a cognitive assessment in 1986, has had no further mental health intervention. He essentially was lost to follow up from all services, and he was diagnosed with Klinefelter syndrome, which could account for his autistic features, his intellectual disability, the concern around learning disorder, and the gait disturbances. So this is just to highlight that these things do occur. So basically, just to summarize, do we do genetic testing in individuals when this is suspected? The answer essentially is we don't routinely do genetic testing for common disorders. Diagnosis is done clinically. However, the YACIPAP suggests that if you have any of these risk factors, such as dysmorphism suspected, intellectual disability, a family history, then genetic testing can be warranted. So I found a geneticist at Gregor who works at Gregor and Red Cross, because I wanted to know what actually happens in our services. And she said that no specific policies are in place. However, the onus is on the clinicians to be concerned or to alert the geneticist about a condition. A genetic clinic happens every Tuesday morning and Wednesday afternoon at Red Cross. Once an illness is suspected based on these risk factors and our clinical acumen, genetic testing can occur. In South Africa, basically, we have access to all sorts of testing for free with the exception of something called a sequencing exome, which can only be done internationally. It's not even done in private. And if a test is not available in the public sector and the geneticist deems it necessary, it will be done for patients via the NHLS. So this just reiterates, I won't go through the slide, but when it's important to be alerted to a genetic disorder, specific things on the medical history, specific concerns on family history, and then physical exam red flags. So, in conclusion, why is this important? Genetics are what makes us unique, what makes us each individual. It explains why we look like our family members and that why certain diseases run in our families. It can also help us learn about our family health history to ensure that we stay healthy. In other words, it's got a preventative approach with certain things like diabetes and hypertension. It can guide treatment and enable access to specialized genetic services, such as those that we have at Red Cross. It also importantly allows for counseling to occur, and let's not forget that prognostic information can be very empowering to individuals. So, as mentioned, another important reason is that genetic syndromes may, in the odd occasion, manifest in a more general psychiatric presentation, which is what happened to the patient that I saw that presented with psychosis and ataxia, and it turned out that he had Klinefelter's. So lastly, I'm just, as a disclaimer, I know some of my slides may seem a little bit insensitive to those suffering from genetic anomalies in terms of showing pictures of them, but just to say that it was done for the purposes of our learning, and that there is an organization an NGO run by a former fashion photographer called Positive Exposure, which aims to highlight the beauty in diversity. So it's a cool website for us to check out. And then lastly, just thanks to Prof de Vries for his guidance. A shout out to Dr. Tina Wessels, the geneticist. All the information is available on the Yakapap free website. And then also to my audience, thank you. And then to open the floor up for any questions. Prof, I think you're muted.
Firstly, applause to you. Secondly, if you can stop sharing, then at least we can just have a big screen of people.
Oh, I'm trying to.
Just the share screen button. Can you stop? I might be able to stop you. Yeah. There we go. Okay, great. So we have 2 minutes left. So let me see if anybody, I see Sylvia already has raised a hand. Well done, Sylvia. So if you want to show yourself and ask your question, that would be great.
Hi. I actually know Ilhaam from last year. And she knows I'm sort of actually more interested in neuroscience than in psychiatry, and one of my chief interests is looking at the sort of polygenetic things and how they result in changes in the physiology in the brain. So I just want to thank her for looking at this, and these are most of the syndromes that we see are very clear sort of genetic things, and especially the sort of, she talks a lot about the karyotype, which is these sort of, you want to sort of on a macro level where you can easily see just with looking at the chromosomes under a microscope what the changes are, but and that's very helpful for clinical practice, but these things are very rare, and so often in medical school we get overwhelmed when looking at them and just sort of forget about them until we see them in clinical practice. So I feel like there's quite a distinct sort of separation between these syndromes that we see and looking at the genetics in terms of a pathogenic, sort of the pathogenesis of psychiatric disease. Sorry, it's not a question, it's more sort of
it's a comment, right? So
statement on it.
No, it's great. It's great to hear that. great to hear that you enjoyed Ilham's presentation about it. And I think you're right. One comment, most genetic disorders that we see these days are actually not big abnormalities like that we would see on a karyotype, but are point mutations. So there might be tiny little changes that we need sophisticated testing for. But just really the broad comment that all genetic disorders, whenever somebody comes into any mental health clinic and they have a known genetic condition, any of them and all of them are risk markers for a range of neuropsychiatric abnormalities and a range of neurodevelopmental difficulties. And so I think the one takeaway lesson for all of us from this is when I see somebody, they have a much higher risk of autism, of ADHD, of psychotic disorders, of anxiety disorders, of scholastic, of intellectual disability. So I think that's a really important message. Second one, and Sylvia, don't think that neuroscience and psychiatry are separate. They can be the same thing. There's a society that some of you might be interested in called the Society for the Study of Behavioral Phenotypes, SBP. And that's an international group of people who are specifically interested in these neuropsychiatric and neurodevelopmental manifestations in people with different genetic syndromes. And so there's a large community of people who do research in exactly this field of research. I see that there's 2 more hands up. Let me check. Tikus, do you have your hand up?
Thank you very much for the nice presentation. One of my questions is already answered. My other question is regarding the legal and ethical issue of this epigenetics and genetic psychiatry in counseling, educating parents and like that. The other thing is when I come to my context, usually we don't advocate to educating parents, particularly illiterate parents, regarding the genetic transmission of the disorder because of the high rate of stigma and discrimination in the community, and how can we handle that unless it is obvious with the presentation.
That's a good,
this is my question.
Interesting comment. Who would like to? Ilham, I'll let you comment on that one first, because that's you raise very interesting ethical challenge.
Yeah, so I think we're lucky in the sense, Tikus, that we do have counseling services available. So we sort of send them to the experts because it's a specific genetic counseling services tailored to providing this service. So pre-test counseling, as the law, is always done prior to testing happening. But I do understand in settings where that facility is not available, I think my answer would just, as we would for any psychiatric illness, I think that it is stigmatizing, it's a difficult thing to do, but I would just use the same principles as I would if I were to counsel a family member of someone with a schizophrenic child or a child with severe intellectual disability. So be open, honest, non-judgmental, empathic, and I think that also recognize that it's a process, that it's impossible to give all the information at one point in time, but also to instill a sense of hope, not false hope, but that there is a variation, or many of these illnesses are on a spectrum of manifestations. That one point in the slide was that knowing is empowering, in a sense that often for parents to actually have a word or description sort of allays anxieties that they may have felt. I think that's just my impression, that they knew something was wrong, but it was never really picked up. Or it also allows them to prepare for the child's future. But that being said, Tikus, I acknowledge it's not an easy thing. I can't say that I've done it myself. So I think maybe someone who has could chip in.
Yeah. So maybe just in the interest of time, I'll just give a quick comment and then we'll wrap up. Doing genetic testing is a little bit like many things we do in medicine. And it's similar to prescribing drugs. It comes with positive effects and it comes with risks. comes with side effects and risks. And we need to, right from the outset, if we're going to have a conversation with mum or dad about the possibility of genetic testing, we need to have the conversation with them about what would be the benefits of doing it and what might be the risks of doing it. And in many of our communities, we know that families are concerned that an association of a genetic label might make their children less marriable, and many people have talked about that might have stigmatization in communities, etc. I do a lot of work with people with different genetic syndromes in the world, and the message that families tell us time after time is, I would like to be able to choose whether I have genetic testing for myself or my child. I don't want medical professionals to make that decision for me. So I would like to have a conversation with you. And then when I choose to do it, more often than not, families choose that they want to have that knowledge, because exactly as Ilhaam has said, it helps them, it gives them knowledge to explain their child or their family's difficulties. It helps them to anticipate future difficulties. It helps them with reproductive decision making. So I think it's an important and a sensitive conversation to have. More often than not, families want to know rather than not to know where these things are available. I'm sorry that we have to wrap up because we've already gone 6 minutes over. So once again, thank you to Ilhaam for a whistle-stop tour through genetics and genetic conditions associated with child and adolescent mental health. The last comment I will leave you with is, we're going to send everyone a note, but INSA, the International Autism Society, will have a free webinar day on the 3rd of June. So we'll send out the link so any of you can register for a 4 hours of webinar on autism-related topics. So please look out for that email, and if you're interested in joining, please do that. So with that, thank you everybody, and enjoy the rest of Friday, and have a good weekend. Bye-bye.
Thank you.